So, as we are gathered in a museum, I thought it was only fitting that I start with this ancient piece from the Museum of Coral Genomics. This is the first fragment of a coral genome ever characterized. It was back in the late 80s by David Mears that I've been privileged to work with. Since then, coral genomics has gone a long way, culminating earlier this year with the publication in Nature of the full genome of Acropora digitifera by Maurice Sato's group and David Mears. The adventure continues with the sequencing of a number of other coral species. And one of these undertakings is the Coral Genome Project that David briefly alluded to, where we have been sequencing the genome of Acropora millepora. So this is a coral with a genome size of approximately 400 millions of letters, which is about 4 million times more than the sequence you saw on that first slide. It's the first eukaryotic genome project undertaken in Australia, and it has this particularity that it is one of the very first genomes to be sequenced using next generation sequencing, the so-called Illumina technique I will present to you shortly. So the traditional method generates read fragments of approximately 1,000 base pairs at a cost of approximately 1 cent per base of finished genome. With the next generation Illumina technique, the cost of sequencing is about 2 orders of magnitude less, but the sequence that you get is also 10 times smaller. So it is easy to fund such a genome project, but the trick is to put these small sequences together. And that's what I've been doing. So most of my work has been to piece these little pieces together with the hope that someday it will help the function of the culprit one day. So this is the plan of my talk. I will spend most of my time telling you about this. And at the end I will briefly touch on some things that we have learned about coral biology based on this coral genome. So we started with the isolation of DNA from coral sperm of a single colony from Magnetic Island. That was done by Eldon Ball a few years ago. And then we feed this DNA in one of these wonderful Illumina machines. And this generates sequences. The sequences typically come in pairs. And there are 2 flavors of sequences. There are paired-end sequences, which are pairs separated by a few hundred letters, and many pair sequences, which are generated by a more complex circularization protocol, and that result in pairs separated by a few thousands of sequences. So now we have to put them together. And it's an interesting game. It's a sort of double-sided puzzle with no picture included, unfortunately. Otherwise we wouldn't have a job. No corner pieces, no side pieces, and a lot of extra junk pieces. So that's why we should play for that 0. And so the traditional way genome was assembled is by taking a read of approximately 1,000 letters, and then finding another read that overlaps. And then by this overlap and extend method, we reconstruct the whole genome. So that's how the human genome was built. And with hindsight, that was pretty easy. But this does not work with the short reads generated by the next generation sequencing technology. The reads are too short to give useful overlaps, and also the sequencing error rate is too high in the first place to trust these overlaps. So the old way doesn't work. So new method had to be developed to deal with these sequences. And to illustrate how this new method works, we will assemble a genome together. Don't worry, it's a very small genome. It's only 10 letters long. So first we have to sequence it. Here we go. The sequences, 2 reads, short reads. And to assemble the genome based on these short reads, somewhat counterintuitively, the first thing we do is to chop them into even smaller fragments. We call these fragments k-mers, or words of size k. So we chop the reads into a number of k-mers, and then we go on to network theory, and we build what is called a de Bruijn graph, which is a graph of overlapping letters, of overlapping words, sorry. So we take our first word, AGA, and we look for other words overlapping. So other words starting with GA. So here we go. We find GA. So we build the first two nodes of our graph connected by an arc. And then we continue by looking for words starting with AT. And we create a new node. And then we look for words starting with TG, GA. So we loop back onto an existing node. And we continue on and on and on like that until we exhaust all the k-mers that we've generated from the reads. So we have now a de Bruijn graph, and we have to unfold it to get the linear genome sequence. But we don't know how to unfold this graph because it is not oriented. So to orient the graph, we look at how the reads flow into the graph. And looking at this flow of reads through the graph, we get a directed graph that can then be unfolded, which results in our genome sequence. So congratulations, you've assembled your first genome. This, the de Bruijn graph framework, is an extremely powerful way to deal with a high sequencing error rate of the next generation sequencing. So imagine we have a bit of genome with two reads and a corresponding de Bruijn graph, and then we have a third read with an error in the middle. This will result in this typical network topology called the bubble. And this bubble can be easily solved using a majority rule and brought back to a linear sequence. So it's pretty simple, the basic idea. Of course, in reality, it's somewhat more complex, and this is not a drawing by my young daughters, but this is the de Bruijn graph corresponding to approximately 0.1% of the whole coral genome. This gives you an idea, 0.1%. So this gives you an idea of the computational complexity that goes into that type of program. So after we build this first genome, we have to assess it to see if it's actually, if we can see if it's correct. And the main way I've been assessing the method that I've developed is using as reference a transcriptome assembly that we've been developing in parallel. So I mapped the genome assembly to the reference transcript. So you can see here you have exons, so gaps. These gaps correspond to exons. The gap here is a sequence that is missing from the genome. And here you have two genome contigs. So this transcript maps to two contigs, you have little gaps, you have inference there. So this is how it should look like. Of course, things went wrong. So what we had in our first assembly is a good coverage of the genome, but the different fragments that you would expect to be collinear were in fact shuffled as a kind of chimera. And this was caused by an intrinsic bias in this next generation sequencing technology. So there are two main types of bias in this technology, and these bias are only associated with the mate-pair libraries that I presented earlier. The libraries obtained by circularization that allow to have reads far away, several kilobases far away from each other. So the first problem is clonality. So we have reads that are not randomly distributed across the genome, but are just copies of each other. And this dramatically biases the coverage of your contigs. The second problem is what I call the shadow library. So you have the large pairs of sequences that are facing outwards, but there is a significant proportion of the reads that are contamination of small sequences facing the other way around. So you can easily guess how these guys would create chimera in the final assembly. So I developed a set of methods to deal with this artifact in the sequencing technology. I won't present the algorithm to you, but suffice to say that when I applied this method to the couple of other genomes that have been published with this Illumina technology, I was able to get at least as good an assembly as what had been published. But when I apply this technique to the coral genome, things are harder. The coral is harder. And the reason why the coral is harder is because it has a very high level of polymorphism, one of the highest level of polymorphism ever described. So as you know, the coral is diploid, it has half of its DNA from its mother and half from its father. But the difference between these two copies is more than 1%. It's a bit like if your dad was a human and your mom was a chimpanzee. No disrespect for your parents. This is how the coral genome looks like. So it might have something to do with the hybridization that John Pendlefield was talking about this morning. So this level of polymorphism is in the same range as the sequencing error rate. So we are in a sort of twilight zone for this next generation sequencing technology. So the fact that these two haplotypes are so different, you either assemble the haplotypes separately, or sometimes you have artifacts like local duplication of short segments. And so I developed a method to identify the polymorphism in the de Bruijn graph. So the basic idea is to look at the topology. So here you have a homozygous sequence, and then two heterozygous, so the two haplotypes. And so the topology of the graph is easy to recognize. And you can also see that this node has a coverage of, say, 2n. So the coverage of the number of reads in average that cover a given base in the genome. And here the coverage is approximately half. So using the shape, the topology of the graph, and the coverage, we can recognize haplotypes and solve them. Of course, sometimes you have more complicated topologies, like around recombination hotspots, you get that sort of topology. Another complication is that the coverage is not just simply the number of reads, but it is a function of the content, the GC content of the reads. So we have to take that into account when we model the coverage of the nodes in the graph. So we have to look at the topology, the shape of the graph, and the number of reads that map on each base in the graph as a function of the GC content in the graph. So finally that worked quite well after we did all that, and we get a genome assembly made of 12,000 sequences, an N50 of 181 kilobases. So the N50 is a size such that half of the genome is on Contigs at least 181 kilobases long. So this compares very favorably with the other published Cnidarian genomes. So as I mentioned, I also assembled a transcriptome in parallel, and all of the transcripts have at least a match in this coral genome, and the large majority of the bases of this transcript are covered. So this is a very complete genome assembly. Finally, we were very pleased to find that unlike a lot of genomes that are published out there, ours doesn't seem to have any bacterial or viral contamination. So this is probably because the DNA, the sperm DNA that we started with was extremely clean. So we released these sequences a couple of months ago on this website, and we're updating from time to time with better assemblies. Now, with this assembly in our pocket, we'd like to know what's in the coral genome, what causes calcification, bleaching, and all that. Well, of course, we don't know. But we can start looking at a few interesting features, and a few easy ways to start looking at the genome is to first look at genes that are more abundant than you would expect, and second, look at genes that are missing based on what we know in other animal genomes. So first, the example of gene family expansion is the expansion of NAFT domain containing genes. So these genes contain a domain called the NAFT domain, it's a typical sequence signature. And these genes are often implicated in innate immunity. They contain things like NOD-like receptors, and as you can see, they are massively expanded in aquaculture. So this complex innate immunity gene repertoire might perhaps be associated with the host-pathogen interaction. Indeed, we know that corals are associated with a diverse and dynamic bacterial flora. So this expansion in NAFT domain containing genes might have something to do with that. Another possibility is that these genes might play some role in the symbiosis, in the interaction with symbiotic. So this is an example of an interesting gene expansion in the coral genome. Now, Another example of analysis that we can do is what genes are absent in a coral, based on what you would expect, well, what we know in other animal genomes. So this is something about the cysteine synthesis pathway. So cysteine is a non-essential amino acid in most animals. And because these animals have a cysteine synthesis pathway that contains a critical gene called CBS, but this gene is missing from acropora. In fact, it's present in several other corals, but acroporids, all the acroporids where we look for this gene, seem to be missing it. So maybe acropora rely on symbiodinium for cysteine biosynthesis. And cysteine might be an essential amino acid for acropora when it's not in association with symbiodinium. So in summary, this coral genome assembly has motivated a number of methodological innovation for the assembly of next generation sequencing data. So this has been in most part released as open source as part of one of the mainstream genome assembly program, which is called Velvet. The fact that a small lab like us is able to assemble a large genome, even if it's a first in Australia, it opens a lot of new opportunity for other small labs who are interested in non-model organisms that big sequencing centers would not be interested in. So I've barely scratched on the surface of a coral genome biology. But also this coral genome sequence is a very powerful platform for comparative studies. So we are currently comparing the genome of Acropora millepora and Acropora digitifera, looking for signature of adaptive evolution in these genomes. And it's also a platform for functional studies, and David and Aurélie will tell you more about the type of functional study that have been undertaken with these sequences. And so I'd like to thank all these people, especially David Miller and Eldon Bolt, who initiated this project, and Thorsten Seaman, with whom I've worked a lot on the bioinformatics side of things.